Today, an FDA approval in acute myeloid leukemia, a priority review designation in non-small cell lung cancer, and exclusive sit-down interviews with Drs. Tanios Bekaii-Saab and Geoffrey Oxnard. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has approved CPX-351 for adult patients with newly diagnosed therapy-related acute myeloid leukemia or AML with myelodysplasia-related changes. The approval for CPX-351, which is a fixed combination of daunorubicin and cytarabine, comes several months ahead of the FDA deadline. It was based on an improvement in overall survival in a phase 3 study comparing CPX-351 with traditional cytarabine and daunorubicin, known as 7+3, for patients with newly diagnosed T-AML or AML-MRC. Results showed that the median OS was 9.56 months with CPX-351 versus 5.95 months with 7+3, representing a 31% reduction in the risk of death. Additionally, the complete response or CR with incomplete platelet or neutrophil recovery rate was 47.7% versus 33.3% for CPX-351 and 7+3 respectively. For CR alone, the rates were 37.3% for CPX-351 and 25.6% for 7+3. At 12 months, the OS rate was 41.5% in the CPX-351 arm versus 27.6% in the 7+3 group. At 24 months, 31.1% of patients enrolled in the CPX-351 arm of the study remained alive compared with 12.3% with 7+3. The median event-free survival was 2.53 months with CPX-351 versus 1.31 months with 7+3. CPX-351 was approved with a boxed warning advising against interchanging the medication with other daunorubicin and/or cytarabine-containing products. The FDA also advised against using CPX-351 in patients with a history of serious hypersensitivity to daunorubicin, cytarabine, or any component of the formulation. In non-small cell lung cancer, the FDA has granted a priority review to a supplemental new drug application for alectinib for the frontline treatment of patients with ALK-positive, locally advanced, or metastatic disease. The agency is scheduled to issue a final ruling by November 30th, 2017. Alectinib was previously approved for patients with metastatic ALK-positive NSCLC who have progressed on or are intolerant to crizotinib. The frontline application is based on results from the phase 3 ALEX and J-ALEX studies. When compared with crizotinib, alectinib improved progression-free survival by 53% in the ALEX study and by 62% in the J-ALEX trial. Alectinib was also found to reduce the risk for progression in the central nervous system by 84% versus crizotinib in the ALEX trial. The 12-month cumulative rate of CNS progression for patients with or without existing CNS metastases at baseline was 9.4% in the alectinib arm and 41.4% for crizotinib. This week, we sat down with Dr. Tanios Bekaii-Saab of Mayo Clinic to share his insight on the recent FDA accelerated approval of nivolumab for the treatment of adult and pediatric patients with microsatellite instability high or mismatch repair deficient metastatic colorectal cancer that has progressed following treatment with a fluoropyrimidine, oxaliplatin, and irinotecan.
We have now accumulated large amounts of data about MSI high colorectal cancer, which occurs in about 4% of patients with metastatic colorectal cancer. It's a higher rate in the earlier stages, but for this discussion, of course, the relevance is metastatic colon cancer, colorectal cancer. So 4% is not a small group. It's a meaningful number of patients given the number of patients that get diagnosed with metastatic colorectal cancer every year in the United States. And so that has significant implications. We knew there was a prior approval with pembrolizumab in MSI-high solid tumors, but the main study included colorectal cancer that were MMR proficient versus MMR deficient, that suggested that MMR deficient or MSI-high tumors have close to 60+% response rate. And these responses tend to be durable, and with follow-up, actually, a lot of them end up being complete responders. And then nivolumab essentially had its own data in colorectal cancer, in the MSI high, showing very similar trends, large response rate, many complete responders, durable responders, very similar profile, and that led to the second approval in the space. Now for nivolumab, the MSI high approval is for colorectal cancer only. With pembrolizumab, it's for all solid tumors. But nonetheless, I think this adds another option for our patients with metastatic colorectal cancer in the MSI high space. Of course, the question becomes, how do you choose between the two? We have data with pembrolizumab, we have data with nivolumab. They're both in MSI high, they both look very similar. So how do you choose between the two? There's no data that compares the two head to head. Historically, they seem to be similar. So it's patient and physician preference, practice preference. Some physicians are more familiar, have worked more with nivolumab, and that may end up being their default. For others, it may be pembrolizumab. I think either way, you will benefit the patients with MSI high tumor with the application of one or the other agent. So this is a welcome addition. It's great to see that happening in a space where we think we can make a big difference for our patients. I mean, responses are just literally amazing. Whomever used these agents in those patients and they've had a response, these responses can last for years and years. And again, I think, like I said, the conundrum is for the practitioner, how do you choose between the two agents that are available to you? And in the absence of head-to-head data, there's really no way to choose between one or the other than patient preference, physician preference, and other factors that may be related to the practice.
We also met up with Dr. Geoffrey Oxnard of Dana-Farber Cancer Institute, who discussed his views on the utility of liquid biopsies in the field of lung cancer.
When I'm thinking about who to send a liquid biopsy on, the question I am thinking for an individual patient is, does their cancer shed DNA? We are looking for DNA that is effectively the needle in the haystack being all of the patient's germline DNA, the needle being the tumor DNA floating around there. And there are certain characteristics that are associated with DNA shed. What we found in our studies so far is the more metastatic sites of disease they have, the more DNA shed. If they have liver or bone mets, more shed of DNA. Versus if this is a lung cancer patient with small lung nodules, asymptomatic, the chance of that being a shedding cancer with a productive liquid biopsy is smaller. And so this is something we each need to really develop a sense about as clinicians, which is in which patients is this tending to be a useful assay, and in which patients is this negative and falsely negative because there was simply no DNA in the specimen for us to assess. If you think about a biopsy, there was a pathologist looking at that specimen telling you, yep, it's got tumor in it, this is a good one for testing. But when you do a blood test, there is no one telling you, we've got good tumor DNA, this is adequate. And so you have to sort of trust, is this a cancer that's shedding DNA? Because if it's negative, you're falling back on tissue testing, the standard, as our backup test because sensitivity is not perfect with these assays.
That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.